Hi guys, welcome back to my channel. Today's video is going to be really interesting because we're going to be doing the IBESS 2.5. So 2.5 is a very interesting and very practical chapter. So it'll be really interesting to do that, and let's get started. So I'm just going to tell you the name and a few little things that you might need to know before starting this chapter. So 2.5 is called investigating ecosystems. So what can be, indulged in this chapter is going to be like this, that you guys see on your screen right now. So you have to, the description of the ecosystems and, talking about how to investigate them. So in that case, we'll have to describe and we'll have to investigate the ecosystems to be able to compare them and make the difference, and know the difference, and you'll be able to monitor them using it, you'll be able to model them, you'll be able to evaluate them, and how the changes are taking place, and you'll be able to measure both of the changes, natural changes that will occur or human impacts as well, rather, and human impacts, because you cannot, you'll be able to. So finally we'll be able to understand the ecosystems better, by investigating them and, actually quantifying them, and, not them exactly, but their components. So, like, what actually exists in the particular ecosystem. So, we'll be able to quantify that. So, there's a few little things that I want to talk about as well before really starting with the detailed chapter. So, let's begin with the next thing that I'm going to tell you guys. So guys, this will be the application and the skills, and how you'll be inculcating these particular topics that you'll be learning, and how they'll be asked, as a question in the exam. So let's just look at them. First of all, the the key terms that can be used are like design, because you can be, carrying like an investigation, or you can be doing that all as well. You can also be told to construct any kind of diagrams and all. So that's another thing that you can be told to do. So please keep these in mind. So construct can be one, design can be one. You can also be told to evaluate in several different. You can also be told to calculate, interpret, and draw. So you guys can quickly go through the questions if you want to. Otherwise, just keep in mind these key terms that can be asked, because you will need to know what they mean. You need to know, what does evaluate mean, what does calculate mean, what does interpret mean, what does draw mean, what does define mean, what does like other of these, I'll say examine mean. So you have to know what they mean, right? That's the key knowledge part of this. Now let's begin with like the actual chapter. So when I start with the chapter, you need to know a few things, a few key points to be able to understand the chapter better, right? So first and foremost and most important is the IB animal experimentation policy. It's the guideline, it's the policy that has to be followed, right? It is also really stringent, in case you guys are wondering. And you have to really be careful before, you design an experiment and you do an experiment, moreover. So let's consider these, keep them in mind when doing experiments, designing them. So what you'll have to consider is a few little things. You have to consider that you should be able to, I would rather say, replace the animal by using cells, plants, or stimulations. Define the experiment to alleviate harm or distress. And of course, reduce the number of animals you'll be involving in that particular experiment. Also, it also talks about, this policy also states, that what an animal experiment cannot involve, cannot cause, or cannot, lead to this. And what I mean by this is It cannot involve any pain for the animal. It cannot involve it being under stress, not involve it being, being damaged or, being hurt in any case. For the health of the animal, cannot involve the death of the animal, cannot involve drug intake, dietary change beyond easily tolerated by the animal. So you have to keep these points in mind before ever doing any IB experiment or any IB animal experiment. So please keep this in mind. And of course, if humans are involved in this, you must have written permissions. Also, for the experiments, you cannot carry out experiments which will involve the transfer of blood-borne pathogens. Okay, please keep this in mind before starting this, because this chapter is going to be all about experiments, all about investigation and evaluation of them. So I have to keep this in mind, let you guys know this is something has to be kept in mind. And you might be asked in the exam that, what particular thing can the animal experiment not involve? It cannot involve any pain to the animal, cannot involve any damage to health. So anything can be asked. So, you should know this all of. It's not that just I'm telling it to you guys because of the fact that, it's there in the particular thing. But you have to know as per your own general knowledge. So I wouldn't concentrate that much on this, but it's something always supposed to be there in the back of your mind. So let's continue. Next is the overview for the data collection. Now let me make this clear here that this chapter is quite big. Wouldn't like to complete it in 1 or 2 videos. Wouldn't like to do that because it's big and I want to give each particular subtopic enough time that it needs to explain. So in this video, we'll be ending it with just telling you and just letting you guys know about the overview of the topics. So I'll just be telling you what we'll be doing. We'll be having quadrats and transacts and what kind of biotic factors and everything. But wouldn't be going in detail. And I'll be having them in detail in other videos, which will be the next part, which will be part 2 of course. Because in part 1, I'll just be giving you guys just about the topics that we'll be studying in the other parts. I hope I made myself clear, and let's just begin. So we'll be talking about where we can collect data from, we can calculate using we can collect it using the quadrats, the transacts, and we can measure either biotic factors or abiotic factors. So we can measure abiotic factors such as marine, freshwater, terrestrial. Also, we can have biotic factors like biomass of particular animal or the species, the productivity, right? We can also be able to catch small motile animals, whether it can be terrestrial or aquatic. We'll be having the keys also, the key takeaways also, and we can also measure the abundance. We'll have Lincoln index for that and the Simpson diversity index. So we'll be having 2 indexes for this particular of measuring abundance. So I hope I made myself clear in what all we'll be doing, and see you guys in the part 2 to follow up with any of these topics in detail. You guys can go and look forward to my part 2, and I'll be more than interested in explaining you guys the topics in detail. In case you guys loved the video, please just like it, and also if any doubt, comment down below. I'll be ready to explain anytime soon. Also, if you guys haven't yet subscribed to my channel, please don't forget to subscribe because it makes a huge difference. 1 subscriber counts, okay? Bye. Have a really nice day and life ahead.